Hello and once again, welcome back to NEET Decoded. Hope you have gone through the previous videos of the NEET MCQ series, and in this video, we will learn about the basic structure of transfer RNA and the process of translation by discussing a NEET question. So let's start. The question is, the first phase of translation is 1. Binding of messenger RNA to ribosome, 2. Recognition of DNA molecule, 3. Amino acylation of transfer RNA, and 4. Recognition of an anticodon. Now, translation is the process of formation of protein through polymerization of amino acids. For this, charging of amino acid takes place. Now, the amino acid molecule is activated in the presence of the enzyme transfer RNA synthetase. This activated amino acid is attached at the 3' CCA end of transfer RNA to form amino acyl transfer RNA complex, and it is an ATP-driven process. So you can see in this picture how charging of amino acid is taking place, and the charged amino acid gets attached to the CCA end or the 3' CCA end of transfer RNA molecule to form the amino acyl transfer RNA complex. Now this is ATP driven, that is the process requires consumption of energy. Now brief overview of structure of transfer RNA is given here. Now the secondary structure has a cloverleaf shape, as you can see in this picture. You can see the similarities here. Now it has 3 hairpin loops, 1, 2, 3. You can see the 3 hairpin loops here. Now the various arms of transfer RNA are the acceptor arm, the D arm, the anticodon loop, the T loop, and the variable loop. You can see here, right? Now, amino acylation is the first step in protein synthesis or translation. The transfer RNA is specific to each amino acid and carries it to the growing polypeptide chain. So the triplet codons of messenger RNA are basically decoded by specific transfer RNA. Here you can see the anticodons here, the position of anticodons. And these are basically recognized by specific codons of messenger RNA, and there is complementary base pairing. So the smallest subunit of ribosome encounters the messenger RNA. As you can see here, this is the smallest subunit of ribosome, and the process of translation is initiated. There are sites in the larger subunit of messenger RNA there are sites in the larger subunit of the ribosome, right? For binding of amino acids. This is the largest subunit of ribosome. Now, during translation, complexes formed of an amino acid linked to transfer RNA binds to the correct codon in messenger RNA through complementary base pairing. So this is the anticodon, this is the codon of messenger RNA, and there is complementary base pairing here. The cytosine combines with the guanine, and adenine combines with uracil. So this is complementary base pairing actually. So the amino acids are thus translated into polypeptide, which is dictated by DNA. So this is the polypeptide, growing polypeptide chain formed of amino acids, and this is dictated by DNA. Why? Because the information in a DNA is transcribed into the messenger RNA molecule. That happens during the process of transcription. So we have the answer. The first phase of translation is amino acylation of transfer RNA, right, which is option number 3. Now for complete set of MCQs and notes covering all chapters, please log into brigademy.com, and request you to like, share, and subscribe to my channel for biology content. And thanks for watching.